Hello, everyone, and welcome to Lecture 7 in our course on general psychology. And this is the third lecture we're going to spend on the brain. It will wrap up our unit on the brain. It will also be the last lecture that will be covered by Exam 1. So the next module will be covered on Exam 2. So the first 7 lectures are what Exam 1 will test you on. So for this unit, we're going to be talking about investigating the brain. How do we measure the brain? And how do we connect what's going on in the brain and how it's built to psychology? So how do we connect the brain to the mind? So first, one thing we can do is measure the structure of the brain. We can look at its various parts, how those parts are connected to other parts, and what happens if that structure is disrupted. We can also measure brain activity, so neural activity, action potentials, synaptic transmission between neurons. We can look at that process and what that tells us about which parts of the brain are involved in which functions. And each of these things each of these bits of knowledge is given to us through a kind of measurement technique. And there are lots of different techniques, but each technique has its strong points and its weak points. There are advantages and limitations to each of them. So we'll talk a bit about what those are. So what we're going to do is talk about the structure of the brain, and especially early on, how scientists inferred what a region of the brain did when the structure was disrupted. We'll also look at some methods that measure brain activity, either at the very small scale of individual neurons or at the much larger scale of billions of neurons. We can measure activity at both those scales, and so we'll talk about how we do that. Well, one of the differences between techniques, not all techniques are the same. Again, they have pros and cons. And so we'll compare and contrast a lot of these different methods for measuring the brain. And then we'll also talk about what these measurements can actually tell us. What conclusions can we draw from a set of data? What conclusions can we not draw? So one of the earliest ways of looking at the brain was by looking at brain structure. It wasn't until relatively recently we could look at brain activity. But people have been looking at brain structure for quite a while now, looking at anatomy for certainly centuries, but even looking at the function of different parts of the brain in living patients for a couple hundred years. And the main way this was done by looking at what psychologically was lost when part of the brain was disrupted. So you lose a brain structure, what can you no longer do, or what do you now have difficulty doing? So we've already mentioned in an earlier lecture Paul Broca. And one of the things Broca is famous for is having a patient who could no longer produce speech. This patient had suffered brain damage in a particular area in the frontal lobe, and that area is now known as Broca's area because Broca is the one who discovered that area is critical for speech production. You cannot speak intelligibly without it. And so that is what is needed to produce speech. There was another scientist named Carl Wernicke who found an area different from Broca's area that was necessary for speech comprehension. So these are two different functions, and we wouldn't have known that if not for these studies of brain damage. So Wernicke, through study of patients with damage to part of their brain, discovered that an area in the temporal lobe near the parietal lobe is critical for understanding speech. So a person could produce speech just fine, but could not understand what other people were saying without Wernicke's area being intact. And both of these areas, by the way, are located in the left hemisphere. These are some of those cases where the hemispheres are not the same in terms of their function. For most functions, they are, but not for speech and language, as it turns out. Finally, another famous individual is Phineas Gage. And Phineas Gage was not a scientist or a doctor like Broca or Wernicke. Gage was actually the patient. And Gage is one of the most famous patients in neuroscience because Gage was a railroad worker and he suffered damage to his brain, and in particular the frontal lobe of his brain, when there was an accidental explosion and an iron rod was driven through the underside of his skull and out the top of the skull. And the way it traveled through the brain uh, damaged the frontal lobe. But Gage survived. Most individuals would not survive an injury like that, but Gage did. He survived, but his behavior was different afterwards. He had trouble keeping a job, for example. He seemed to have had trouble with emotional control, with maintaining social relationships. Uh, and, and these functions, things like planning, decision making, emotional control, seem to be housed in the frontal lobe. And we know that because of patients like Phineas Gage. Now, the degree to which Gage's behavior was disrupted is a matter of some debate. Some people indicate that uh, he was a totally different person afterwards, that he was no longer recognizable. Uh, Other people say that he didn't change all that much, that the story has been kind of sensationalized. And that debate is not important for us here today. But certainly we know that the frontal lobe is involved in certain critical functions, especially higher functions like planning and decision making. Uh, and most of these historical studies of brain structure have been through brain damage, through accidents. Also things like strokes or the removal of tumors. Uh, but brain surgery, including the removal of tumors, but also surgery intended to alleviate seizures, has also shown us a great deal about the importance of various structures in the brain. And so here, the idea, of course, is not that surgery is intended to study the brain. That would not be ethical. We do not remove parts of people's brains as a means of determining what that part of the brain did. Uh, no, these are surgeries that are necessary for other reasons. Uh, and, and through the course of surgery, part of the brain is removed, and the loss of function is a side effect. Again, a lot of these surgeries are an attempt to alleviate epileptic seizures or seizures of other origin. Uh, and so if removing the part of the brain that tends to generate the seizure alleviates the symptoms, that's great. But that surgery also often carries with it the loss of certain functions. And probably the most famous patient in all of psychology is patient HM. And in this picture here on the right, we see on the left an intact human brain. And this is a cross section called the coronal slice. So if you separated the front half of the brain from the back half, and then looked at the cross section, that's the view we're seeing here. The right image is HM's brain. And HM had both hippocampi removed, in addition to some other tissue. And we talked last time about the hippocampus and how important it is for memory. So HM lost both hippocampi, and as a result, he could never form new memories after that. He could remember everything that happened before the surgery, and he could learn new skills, which are a different kind of memory. But he couldn't remember uh, people that he'd met since the surgery, couldn't remember anything that happened to him since the surgery, and so this was a very devastating set of side effects. His seizures largely went away, but his memory did too, his ability to form new memories at least. And so he, again, one of the most famous patients in all of psychology that we learned a great deal from because of brain surgery. Also, there are a collection of patients known as split brain patients. And these are patients that, in order to alleviate seizures, have had their corpus callosum cut or removed. So that is the corpus callosum being the white matter, the axons that travel from one hemisphere to the other. It's how the two hemispheres of the brain interact with one another. It's how they communicate. It's how information is transferred from one half of the brain to the other half. And again, this is typically done to alleviate epileptic seizures. If seizures begin in one hemisphere, they can spread to the other. And when medication doesn't work, people will often turn to surgery or to these split brain procedures so that that seizure cannot spread to the other half of the brain. And it works, but it does produce an interesting set of side effects. And in effect, what the left hand doesn't know what the right hand is doing, to borrow a metaphor. So the two hemispheres are now almost completely independent. They don't share information. And so in this picture here from your textbook, if you present pictures such that one eye or one visual field, to be more accurate, sees one stimulus, in this case a key, and the other visual field, that is the right half of the visual world, sees another stimulus like a ring, that those are now not, those separate pieces of information are not communicated to the other hemisphere. What does that do? So our visual world is split into left and right, and it's an interesting fact of our organization of our nervous system that the right visual cortex processes the left visual world, and the left visual cortex processes the right visual world. Similarly, the right hemisphere controls the left side of the body, and the left hemisphere controls the right side of the body. So there's sort of a crossing over. What does that do for split brain patients? Well, it means that if the key is shown in the left half of the visual world, it's processed in the right hemisphere. The ring, which is depicted on the right half of the person's visual world, is processed in the left hemisphere. So the left hemisphere knows about the ring, and the right hemisphere knows about the key, but they can't communicate with one another. So if you ask the person, what do you see? They will say a ring, because speech is handled on the left side of the brain. And the left side of the brain knows about the ring. The left hemisphere doesn't have access to information about the key, so it can't say anything about the key. And the right hemisphere can't say anything about it because it doesn't have a speech center. And so if you ask, the person will say a ring. If you ask them to point with their left hand to what object they see, they will point or pick up the key, because the left hand is handled by the right hemisphere, the key is being processed by the right hemisphere, and so depending on how you ask the person to indicate what they see, they will choose one object or the other, and they are not aware of both. And the important part here is that speech is lateralized. That is, it is dedicated to one side of the brain, in this case the left, which is why they can say ring but not say key. In a similar experiment, if you show someone with a split brain procedure what's called a chimeric face, that is a composite image where the left half is one person and the right half is the other, then what they report is something very similar. If you ask them to say who they see, they will indicate whatever person is on the right half of the image. So in your book, in this case, uses Brad Pitt and Leonardo DiCaprio. DiCaprio is on the right, and so the person says Leonardo DiCaprio. If you ask them to point between two images as to which person they see with their left arm, they'll point to Brad Pitt, because again, the left arm is controlled by the right hemisphere. Brad Pitt is seen by the right hemisphere, and so that is what the person will indicate. And so this split brain procedure creates this really interesting side effect where half of the brain doesn't have access to what the other half has access to and can't control what the other half controls. And so it's almost like having two individuals within a single patient. So as I mentioned early in the history of neuroscience and psychology research, looking at brain injury and surgery and the changes that occurred in behavior as a result were really the only way to understand the brain. But more recently, and this is within the last 80 years or so, we've been able to look at brain activity, that is to measure action potentials one way or another. And so that gives us new understanding of how the brain works. As we know from our unit on neurons, neural activity comes in the form of action potentials. Now, there's also chemical activity when neurons communicate with one another, but we know about this electrical signal that happens within a neuron when it's firing, when it's active. So one way to measure these action potentials is through an electroencephalograph. So electro, because it's based on electricity, encephalo is the word for brain, and then graph, or record, or writing, literally. This is often abbreviated EEG and looks like this here on your right, where you have a series of electrodes that are placed on the head, usually on the scalp. So this is a non-invasive technique. It does not involve breaking the skin or opening up the skull, and that's usual for EEG. There are sometimes patients in hospitals where if you're trying to figure out where an epileptic seizure is starting, you will actually have EEG electrodes inside the skull, but that's relatively rare. Typically, they're placed on the scalp, and this sort of research is done in lots of psychological laboratories. It doesn't require a special patient. This technique is very fast, and what I mean by that is you can acquire measurements hundreds, perhaps even thousands of times a second, which is about the same time scale on which neurons fire. So neurons can fire up to 1,000 times a second, and you can use EEG to look at that activity within that kind of time frame. So it's very fast. That's one of the advantages of EEG. One of the downsides is you're really just looking at the outside of the brain. You're looking at the outer surface of cortex, which really, which certainly limits you in terms of which parts of the brain you can measure. And when you are measuring, it's not spatially very precise. You're not measuring the activity of a single neuron. You're measuring millions or even billions of neurons at once, which may be what you want to do. If you want to get a picture of global activity across the brain, EEG is a great technique for that. If you want to know what one individual neuron is doing, you can't do it with EEG. For that, you need what's called electrophysiology. So it's still based on measuring electrical activity, but here, an electrode or multiple electrodes are actually inserted into the brain. So this is an invasive procedure. It's rarely done in humans, unless there needs to be an electrode there anyway for therapeutic purposes. This is typically done in animals, or with individual slices of brain that have been removed. And this is what it looks like. So you can usually see this under a microscope. The kind of triangular part of the image over here on the right is the electrode itself. So it's a very, very thin, usually glass tube that can be inserted into an individual neuron. And this technique is how we know about action potentials, what they look like, what they're based on. It is knowledge that we have largely thanks to electrophysiology. And this is a much more precise technique. You can still measure things very fast, but you're now looking at just a single neuron, or maybe a group of neurons. And this image here at the bottom is the sort of data you get from electrophysiology. You can see these upward spikes. Those are action potentials. So you can measure individual action potentials from a single neuron. But again, this is just one technique. This one happens to focus on electrical activity from neurons. Another technique that's common now is brain imaging. So imaging, as the name implies, means taking a picture of the brain. Some imaging techniques emphasize brain structure. So these are things like computerized axial tomography, which we see here on the left part of this image. This is often called a CAT scan or a CT scan. And so what this is basically a three-dimensional X-ray. So we've probably all had an X-ray before in one form or another. And CT uses the same technology. It's passing X-rays through the body. Certain tissues absorb X-rays more than other tissues do. And so whatever gets through is then exposed on film. And then we can see which parts of the body are more dense than others when it comes to X-rays. And that's what a CT is. And so you can look at the differences between different parts of the brain there. Which parts of the brain are intact? Where is a tumor, perhaps? Another technique is magnetic resonance imaging, or MRI. Many of us have probably had an MRI. It's shown here on the right. There are multiple ways of getting an MRI image. So in this one, we can see that air shows up as lightly colored. But we can also see the brain. And we can see the difference in what's called gray matter, the outermost part of cortex, and white matter, which is the inner part of cortex. And the reason it's white is because of myelination. So the inner part of the cortex is filled with myelinated axons, white matter. These are both structural techniques. So you can look at the structure of the brain, but it doesn't tell you what parts of the brain are active. Another imaging technique is diffusion tensor imaging, or DTI. And we see this multicolored image here below our CT and MRI images. This is a very different sort of image. It doesn't look like a brain. What this technique does is trace axons from their origin to their destination. So you can figure out which parts of the brain are connected to which other parts. And this is a relatively new technique. X-rays and CT have been around for decades. MRI has been around for a few decades. DTI, relatively new. So this lets us trace that white matter to figure out where a particular set of neurons start and where they end up. So you can see which neurons are talking to which. So those are all structural techniques. They tell you how the brain is built, how it's connected to itself. They don't tell you which parts are active. For that, you need functional imaging. And we were talking about electrophysiology and EEG. Those are also functional techniques. They tell you about brain activity. And they're relatively fast. But they can only tell you about a few neurons, or they tell you about a lot of neurons, but only on the surface of the brain. Functional imaging is slower. You're only getting an image of brain activity every couple of seconds. But it's thorough. You can image the entire brain. So that is the trade-off that's often made between something like EEG, which is fast, but not comprehensive. You can only see parts of the brain and only in very broad strokes, versus functional imaging, which is slow, but you can see the entire brain. So it's just up to the researcher which technique he or she wants to employ. One of the earlier techniques for functional imaging was positron emission tomography, otherwise known as PET. And you may have heard of a PET scan or even had one before. And the way PET works is by injecting a small amount of radioactive substance into the body and then seeing where that substance goes. So usually in the case of PET, at least for neuroscience and psychology, it's radioactive sugar. And of course the brain needs sugar to operate, and the more active a part of the brain is, the more sugar it needs. So you can see which parts of the brain are more or less active based on how radioactive those parts are. You can detect, using a PET scanner, radioactivity, where those molecules have gone. And so you can see where those more active parts are located based on which parts are emitting more of that radioactivity. One of the drawbacks is you have to inject this radioactive material, which is not harmful in small doses, but you can't have a whole bunch of PET scans within a small period of time for that reason. Later on, a new version of magnetic resonance imaging was developed called functional magnetic resonance imaging, or fMRI. So that is a technique that looks at oxygenated hemoglobin. Hemoglobin is the molecule that carries oxygen in your blood. And what's interesting is that an MRI machine is essentially a giant magnet. And this magnet is able to detect small changes in magnetic fields within your brain based on whether hemoglobin has oxygen bound to it or not. The hemoglobin molecule in your blood changes depending on whether it has oxygen bound to it. And so we're able to detect that all over the brain using an MRI machine. And when an area of brain tissue is more active, it needs more sugar, as we saw in PET. It also needs more blood. It needs more oxygen. And so when an area of neurons becomes active, when they're working, when they're firing action potentials, they need more blood. And oxygenated blood gets flooded to that area. So the brain controls where its blood supply goes. and more active areas get more blood. And so we can detect that change in the magnetic field of the brain by using fMRI. And this is a very popular technique now in psychology for looking at brain activity as a person performs a particular task. PET is now not used so much, uh, but it was used for the same purpose. You'd have the person engage in a task and then look at which areas of the brain were active, whether you were asking them to remember something or complete a math problem or listen to speech. These are all different psychological functions, and by looking at brain activity, we hope to understand which parts of the brain are involved in which functions. As we'll see here in a minute, we have to be a little careful about how we interpret these data, though. An even newer technique for understanding brain function actually manipulates the brain. So when we're using EEG or electrophysiology or a structural imaging technique or PET or fMRI, we are measuring the brain. We are not changing anything about it. But with this new technique, we can actually manipulate the brain. And that lets us say more about cause and effect. If we recall from a few lectures ago, we have to be careful not to mistake correlation for causation. And the way we get around that issue is through an experiment in which we manipulate one group of individuals and not another. And so a technique known as transcranial magnetic stimulation, or TMS, allows us to do that. And what this technique does, and this is a real-life person depicted here, this technique uses a magnetic field to either activate or deactivate a portion of the brain. Depending on how the machine is used, it can do either. So it can induce activity, it can make action potentials happen within neurons, or it can deactivate a region of the brain. And you can see differences in behavior afterwards. So what you usually do here is measure someone's behavior and then stimulate or deactivate the brain using TMS, and then look at their behavior again. Or you have two groups of people, one of whom undergoes TMS, the other of which doesn't, and you look at differences in how those two groups behave. But either way, what we're actually doing is changing brain activity. So we can see what activating a region does. We can see what shutting a region down does. So now we can get more accurately at cause and effect relationships between brain activity and psychological function. There are some ethical considerations here. For a while, it wasn't certain whether TMS caused permanent changes in brain activity. We're pretty sure now that it doesn't. But of course, when you're changing someone's brain, when you're manipulating the brain, you have to be a little bit careful. If you're shutting down part of the brain, and then later on after the experiment, the person behaves in a way they otherwise wouldn't, especially if it was a dangerous behavior, the experimenter is in a sense responsible for that. So which is why when a person undergoes a TMS experiment, part of the procedure is they have to hang around after the experiment for half an hour or so until we can be confident that the brain activity is returned to normal. So when you're manipulating the brain, even in as temporary a fashion as TMS is, there are still some ethical considerations to be made. Okay, so we have our data. We've done our brain imaging study. What do we do with those data? How do we interpret them? Well, so first, let me say that brain imaging has indeed confirmed a lot of the functions that previous studies of brain structure hypothesized. So if you have someone in an fMRI scanner and they're producing speech, if they're speaking, you can see activity in Broca's area. If you play speech to them and they have to process human speech, you'll see activity in Wernicke's area. There are decision-making tasks or tasks that require self-control, tasks that employ the frontal lobe, the part of the brain that Phineas Gage had lost. When you have people perform these tasks while undergoing brain imaging, you can see activity in the frontal lobe. And of course, not all brain imaging is used to just confirm previous hypotheses. It's also given us some new insights. So looking at consciousness, for example, looking at people that are awake versus asleep versus under anesthetic, and looking at brain activity has shown us perhaps some of the underlying structures involved in consciousness. Things like reward processing. How do people process when good things happen or when bad things happen? How do they adjust their behavior afterwards? What role does surprise play in processing those sorts of events? So brain imaging has contributed knowledge in those fields as well. But we have to be a little careful when we're talking about imaging data. Uh, and the reason for that is that imaging and things like EEG or even electrophysiology are correlational. That is to say, you're observing some behavior, you're observing brain activity, and you're inferring some relationship between the two. And especially for complex behaviors, you might see activity in lots of different areas. If you have someone read a word aloud and you see activity in several areas, that does not necessarily mean that those areas are the areas of the brain responsible for word reading. Think about all the things that have to go into reading a word. You have to see the word, so there's visual processing involved. You have to recognize the letters as symbols representing sounds. That's a psychological function in and of itself. You have to recognize the meaning of the word. That's going to involve an association area. And then you have to produce the sounds. You have to move your mouth, lips, and tongue to produce speech. And so all of those different functions are involved in reading a word. So again, we have to be careful when we see a pattern of activity, not to say that all of these areas are involved in the most complex version of that function. Now you can design clever experiments trying to get around that, trying to say, well, these areas are involved in speech versus these areas are involved in just visual processing of words. So you can carefully design your study to try and address that issue. But it's very difficult to address entirely. And again, the basis of this is that it's difficult to separate out cognitive functions. Now, lots of behaviors involve multiple functions, especially complex behaviors. Think about social interaction. So it involves visual components, auditory components, involves memory of interacting with the person before. It involves emotions. It involves decision making. What action should I exhibit? How should I respond to this person? So all of these different things get built into social interaction. So if we do an imaging study where somebody's engaged in a social interaction, we have to be very careful not to overinterpret the data. And then finally, imaging studies almost always reflect upon a sample. So we know how the average person behaves. We know what the average person's brain activity looks like when they're exhibiting a behavior or thinking about something. It doesn't really tell us much about the individual. And your book makes the point that, especially for things where the stakes are high, like a trial, that we shouldn't necessarily use brain imaging data to say something about the individual, especially for things like lie detection. So there's some evidence we could use brain imaging as a lie detection device. But again, it only works, as far as we know, at the level of the population. We know what the average person's brain looks like when they're telling the truth or lying. But that doesn't necessarily mean that finding applies to all individuals. So you have to be very careful taking a generalized conclusion and applying it to a single person. All that being said, brain imaging has proved to be a very powerful and accessible technique, and we're learning a lot about the brain thanks to brain imaging, thanks to EEG, thanks to electrophysiology. So the fact that there are correlational techniques is not the end of the world. Okay, that will do it for our lecture on methods in investigating the brain. Next time, we'll actually start a new module on sensation and perception. So how do the senses work? We'll talk about physiologically how the senses work, how we get information in general from the environment to our brain. In particular, we'll talk about vision next time. So how does the human eye turn light into action potentials. How does a complex stimulus out there in the world become patterns of neural activity? And then once that information gets to the brain, how does the brain interpret our visual world? How accurate is that interpretation? How much processing is involved? How do we interpret a stimulus and all the visual properties of a stimulus? So we will talk about that next time, and I will see you then.